Good afternoon everyone, my name's Andy Yates and I'm the Chief Scientific Officer at Artelo Biosciences. Artelo is a NASDAQ listed company and is a clinical stage biopharmaceutical company that's exclusively focusing on treatments that target the endocannabinoid system. Because we're listed, I just need to go through the forward-looking statements. Some of the things I'm going to talk about today are related to research and development, which have unknowns with them. And if you want any further details on the risks that we're absorbing, please take a look at all of our filings, which are on our website. So the endocannabinoid system is something that we've all got in this room today. We were all born with one. And it's a diverse, promiscuous system of receptors and neurotransmitters that's located throughout the body and in the brain. And because of that, I think it's a very exciting area to be in, because the statement at the bottom in orange is a statement from the NIH, which effectively says that it has the ability to target diseases in a number of different areas. And I draw your attention on here to obesity, metabolic syndrome, cachexia, and some of the anti-inflammatory conditions. So our portfolio consists of 3 different drugs, and each of them are at different stages of the development cycle. The top one, which is 2713, has been into the clinic and is about to go back into the clinic this year. And we have 2 compounds that are heading towards the clinic in IND enabling stages. We have a fatty acid binding protein inhibitor, which is designated 2612, and a novel formulation and composition of matter of CBD, which I'm sure some of you have come across. It's widely available, but we've made this a pharmaceutical grade, and we've overcome some of the hurdles of working with CBD related to intellectual property and its pharmaceutical characteristics. The thing that sets, I think, Artelo apart from some of the other companies in this space is that we're targeting some of the bigger indications that we can go after. Because we've got either intellectual property granted or pending for all of our molecules, we can invest or think about partnering to invest in the conditions that are actually going to be big revenue drivers, as noted on the right hand side of this slide. Other companies that are working with cannabinoids are somewhat restricted, if they don't have the intellectual property, on having to stick within orphan indications, and you've seen that with some of the companies out there, simply because they don't have composition of matter to work with. Speaking of composition of matter and IP, we have 2 molecules that we have licensed. 2713, which is the cannabinoid agonist, was licensed from Neomed, which was originally discovered in the laboratories of AstraZeneca. And the fatty acid binding protein was licensed from Stony Brook. And our CBD co-crystal was developed internally by Artelo scientists working with Albany Molecular Research to deliver the science behind that program. So we've got worldwide exclusive licenses to the IP for the top 2, and we've got patent pending on the co-crystal. We're expecting that in the very near future from the US Patent Office. So let me introduce you to the first of our programs, 2713. Cancer, anorexia associated with cancer, is a condition that affects 60% of patients with advanced cancer. It's debilitating because it takes away their robustness for future cancer treatments, but it also it affects their quality of life and means that their ability to live well with cancer, which is a phrase I've come across recently from speaking to the key opinion leaders, is reduced significantly. Even though there's no licensed medicines available in the US or indeed Europe, the market is still quite large, at about $1 billion in both US and Europe, and that is widely made up of medicines that don't have a license but are widely used, such as steroids, anabolic steroids, and other appetite stimulants. There's two things that I'd like you to take away from this slide in relation to how ART 2713 may work. We know from cannabis users that they experience an increase in their appetite and their drive towards high calorific food substances, and so we know that cannabinoid agonists stimulate appetite. What you'll see on the next slide is that our cannabinoid agonist is peripherally selective, so it works mainly in the body and not so much in the brain, and we therefore avoid side effects. But we do know from a lot of research done that there's a strong gut-brain axis, so it's not surprising that our molecule agonizes the cannabinoid system in the periphery, and that can have a feedback mechanism on the brain. This is the molecule in question. It's completely structurally distinct from any of the plant-derived cannabinoids, and as I said, it was designed in the laboratories of AstraZeneca by medicinal chemists, and it was designed specifically to be peripherally selective. So you can imagine that why that was done. It's to avoid some of the centrally acting side effects that are commonly associated with cannabis-based medicines. Don't take my word for it. This is the PET scan of a labeled molecule in the monkey. So you can see from the heat map here that the drug targets everywhere except for the brain. And on the right hand side is a table of the side effects that were seen in a 12-day multiple ascending dose study conducted in healthy volunteers at a dose of 250 micrograms. And you can see that the side effects rated by severity was comparable with the placebo arm in that study. One of the reasons we think that this program from a clinical perspective is somewhat de-risked is because we have evidence from the 12-day MAD study of dose-dependent, or actually plasma concentration-dependent weight increases seen at every single dose that was tested. This was highly significant, over the P of less than 0.0001, and we are about to go back into the clinic in patients with anorexia associated with cancer and test that this hypothesis, or sorry, these results are seen in those patients with that unmet need. So we're running a combined phase 1, phase 2A study, all in the UK. We're currently, we've had regulatory interactions, we're producing clinical supplies, and we have secured sites within the UK. We're expecting to start in the very near future, and we're hoping to have data by the end of this year from at least some of the subjects. Next is a very interesting program, fatty acid binding 5 inhibitor, ART 2612. This is a program that was born out of Stony Brook University and had received, prior to Artelo becoming involved in a partnership, significant NIH money. We got interested in the program because of its ability to chaperone endocannabinoids, so the body's own cannabinoids within the cell. We've become even more interested in the program subsequent to finding out that it's not only endocannabinoids that fatty acid binding protein chaperones around, but other fatty acids, particularly those associated with the fatty acid metabolism cycles of cancer cells. So you can see here an example of fatty acid binding protein carrying fatty acids towards the PPAR gamma target within the cell, which has an output of VEGF, which, as many of you will know, is a pro-cancer growth factor. And I wanted to share some data with you today relating to the fact that fatty acid binding 5 protein is well correlated within cancer systems. So graph A is showing the effects of tumour growth when you have fatty acid binding protein present and when you knock it out using genetic silencing. And you can see there a clear difference between the tumour size when you knock out fatty acid binding protein 5. And graphs B, C and D are all showing the correlation between the presence and concentration of fatty acid binding protein in the tumour grade in B, the triple negative breast cancer status within breast cancer types in graph C, and the prognosis of prostate cancer in graph D. So in all cases where fatty acid binding protein presence are high, you have a worse prognosis with the cancer. So we went to the University of Liverpool and we used one of our lead compounds, compound 26, and we put that in an animal with castrate-resistant prostate cancer. And you can see there from the graph that by using an inhibitor of FABP5, we were able to show very similar effects to what you saw on the last graph. Take that a step further and add in fatty acid binding 5 inhibitors on top of taxanes, which are the standard of care in prostate cancer, and you can see that we can get synergy with the taxanes. Only yesterday, the National Cancer Institute of the US awarded Stony Brook $4 million in a grant, which was a very unusual R01 grant, and that was picked up by investors and the market as a very positive validation of this technique by somebody very established like the NCI. So we're very pleased with that grant, and that's all flowing into our worldwide exclusive licenses. And finally, but not least, the CBD co-crystal. So CBD is a molecule which has a wealth, a treasure trove of data associated with it. The issues with CBD is it doesn't have any composition of matter patents, and it's not a particularly good molecule to formulate with. So Artelo scientists overcame that by forming a co-crystal. Co-crystals are where you take CBD, you mix it with a co-former, in this case TMP, and you create a very stable crystal lattice. In this case, we have increased the melting point of the crystal, and we have abolished any polymorphism that you see with CBD, which in themselves are significant movements in the molecule. And that has led to us being able to file patents, worldwide patents, with this molecule. Co-crystals aren't new to the drug industry. On the bottom of the slide are examples of 2 blockbuster drugs that are co-crystals, and all regulatory authorities recognise this as an approach to overcome issues with drug development if the molecule requires it. The real benefits here is it allows all the known data and potential benefits of CBD to be taken into larger indications. And in Artelo's case, we are targeting post-traumatic stress disorder and inflammatory bowel disease as 2 of our lead indications for this molecule. A little bit about the company. It's staffed by experienced pharma execs with a long history of knowing what good and what finished looks like. Steve Reich, our Chief Medical Officer, is an oncologist and has experience in running anorexia and cachexia trials. Doug Blaney, previous president of ASCO, sits on our board, and Saoirse O'Sullivan has a 20-year history with the University of Nottingham in cannabinoid pharmacology. We're traded on the NASDAQ, and we have a market cap which went up quite significantly yesterday on the NCI's data. So these slides are old, roughly just under 10 million today. And whilst we had a very busy year last year, we're expecting even more news this year, and you can see there some of our key milestones coming over. We're expecting action by the US Patent Office on our co-crystal. We're going to start our clinical trial in anorexia in cancer and have data available by the end of the year if things go our way. So I'll leave you with this slide, which just gives you a reminder that we've got some short-term news flow, which is very interesting, but if you think about our long-term potential with what I said about the endocannabinoid at the system at the beginning, I think there's a huge amount of potential in the portfolio that we have and the portfolio that we're developing. And with that, I'll take any questions if we have any time.